Hello, boy-leos and girlios. I'm going to show you guys a DNA model. Alan Mock said, hey, I don't know what to do about this. Could you please show us an example? So I came home after work today and I put together a quick little example for you. So bear with me for a moment and I will show you this. Then I'll go into the lesson that you guys have to complete today. So let me see. All right, so the DNA model that I have for you guys that I created, it has a title, it's nice. I did a virtual one, not an actual one. And this is my model. I think it's quite attractive. Now, I think that probably you guys are a little better with your artwork than I am. Ooh, went jump to the key. I think you're probably going to be a little better at this, but let's see. What do I have? So I do have a 5N. I know that because there's a phosphate group here. 5N and 3N are the opposite of one another. So in the 5N, you have this phosphate group that's going to be extending here. It's over here. It's anti-parallel, so they're going different directions. You can see, I know it's going different directions because of the orientation of the nitrogenous bases. You can see they're upside down here, or at least the opposite of what they would be. The C and the Panasonic is here attached to the deoxyribose, and the C is here is attached to this bond here. Now these are all covalent bonds, and you don't have to. These are phosphodigestive bonds or digester bonds. You don't have to know that necessarily. You just know these are all covalent bonds, and those are indicated by these solid lines that are connecting them. But anyway, 5N, 3N, 5N, 3N, anti-parallel, opposite, the backbone is the opposite on either side, and the orientation of my nitrogenous bases is also the opposite. Now, you can see that I have symbols for the hydrogen bonds. You do have to have a key for those, and I'll show you the key in just a moment. So these are going to be 3 hydrogen bonds because this is guanine and cytosine. This is adenine and thymine, and so they have 2 hydrogen bonds. And essentially that's it as far as the bonds are concerned. The gumdrops, I thought it was kind of funny. So because it's a sugar, it's a carbohydrate, so it's deoxyribose. These are indicated by these gumdrops. Phosphorus is a solid. It doesn't have a gaseous component, so I thought I'd make it a little kind of funny and make it as a rock. Phosphates get stuck in rock. So we'll learn that when we get to ecology. But there we have our virtual model of DNA. Pretty straightforward. Chargaff's rule is adhered to. Guanine and cytosine are bonded, and adenine and thymine are bonded together. So there we have, and I put 14 nitrogenous bases and 7 pairs. So do at least have 7. I mean, some of you guys are probably gonna go overboard. You're gonna make it helical. I couldn't figure out how to make it 3D. I'm a 2D man, so 2D is fine if you need to do. If you can show me 3D, I'll be pretty impressed by that. But this is just to get the point. You do need to know all these little facts. They're going to be tested. They're going to ask you questions about this. Hydrogen bonds are weak, and these covalent bonds are not weak. They're strong. So whenever they do break, whenever you're going to have an enzyme that breaks these helicases, it's going to split these hydrogen bonds, and it's relatively easy for them to do that. These bonds are not broken during replication, but we'll learn that next week. Here's my key, phosphate group, nice little rock, deoxyribose. We know it's a sugar, OSE. It's a sugar. Guanine is an energizer battery. Duracell, hold on. Oh, shoot. Duracell is my cytosine. I wish I'd turned that around. It's backwards, but I guess it's okay. CVS battery for thymine. Cytosine is going to be a Panasonic. So why did I have cytosine here twice? What's wrong with me? Guanine, cytosine. This is supposed to be adenine. I made a mistake. Oh my God. Let me fix that. What is wrong with me? We'll fix this before you present. Okay, we're okay again. Life's better. Then my hydrogen bonds. I use buttons because I thought, they put these together. So that's my little clever take on this. Please don't copy me. Do your own little thing. Have fun with it. Don't be so weird about it. Okay, so what are we going to do today? So let me show you. Oh, I have a way, and I'm not going to say how, to know whether you pulled this from somewhere else or if you created it yourself. Cannot give you my trade secrets. So let's go to Schoology. That's where everybody wants to be on Schoology. Oh, yeah, that's where I want to go. Schoology for me. Go to Schoology. I don't want to take a tour. I want to log in. Now that I'm logged in, let's go to your class. What? Wrong course. Go to class content. Go to genetics. And I'm going to go right now to the asynchronous test. Take your exit ticket for today. It's pretty straightforward. I really just want to get to, even though I don't get to see you guys, and it really does make me sad, I don't know as much about you as I'd like to know. So really what you guys have learned is how we came to understand what DNA was, that it was a molecule for heredity. It has a huge implications. Obviously, I can't get into all of those because I'm kind of stealing your thunder. There's a lot of ways that people are familiar with it. There's a lot of BS surrounding it too, but it's a pretty important molecule, man. I mean, it explains everything that every emotion you've ever had, everything you've ever seen that's alive is all encoded in this 4 nitrogenous bases that comprise DNA, which is pretty incredible to me. Nevertheless, so you have this pretty straightforward response here, and I just want to know, it's subjective. Let me see if I can make this a little larger. Subjective prompt. And it has a couple of reasons that I'm asking you this. One, I would really want to work on these command terms. So go to the library, make sure you know what explain is, describe is. Those command terms are very, very commonly used. And I want you to use these command terms to explain why the discovery of DNA, that the discovery of DNA was a molecule of heredity was important in discovery. Why was it an important discovery? Did I word that correctly? In your words, explain why the discovery of of DNA, discovery of DNA. Oh no, that was right. Discovery that DNA, because they thought it was proteins, that DNA was a molecule already was an important discovery. So why was it important that they understood this? They, meaning our ancestors, people that preceded us. That's it. I just really want to know, why do you think it's important? Why do you think it's important? And it is important. So why was it important that they knew the difference between protein and DNA? DNA is, as a structure operates by a set of rules, is a structure operating by a set of rules. Yeah. Describe a set of rules that you live by, which would identify you as a person. So DNA operates by a set of rules and identifies an organism as to what it is. But what rules would identify you as a person, like individually, and maybe even as a group? So do you have a set of rules that you live by that are unique to you, as well as are they, are those set of rules also put you into a particular group? So me as an environmentalist and somebody who's very, 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 very centered on the ecology, my friends and I, we all live by a certain set of principles that we're very conservative in how we utilize resources. I pride myself in not utilizing more than I need, even though I do, but I try to curtail as much as possible. I drive infrequently. Those are some rules I kind of live by. If I can walk, I walk. If I can't, then I will drive. If I do, I drive very efficiently as much as possible. And I also want to know whether or not you feel like these groups, who are these groups that you belong to? That kind of, included in that group of people that live by that code, but there's other things about me that are important, and rules and dogma that I live by that aligns me with certain groups of people, but also kind of sets me apart. So what is it about you that I don't know. Try to do something, include something I don't know, something about you. And you do this, you hit all three of these command terms, and you give me a little information about why you think DNA was important to understand something about you as an individual, and then explain why that matters to you as an individual. At least those rules, they really kind of define you as a person. And then also explain why they could also include you with a like-minded group, and let me know a little bit about yourself, and boom, you get a 100. Do make sure you include your date and name on it. I'm going to have it as a document that you're going to open, and obviously it'll open uniquely to you. But when I'm grading it, it's just kind of tough. It's all going to be, it's going to say importance and rules, right? So if I see your name, I can just drop it right into the grade book really easily. Again, if you need me, my phone number is 817-714-2886. You just call. Finally, if you have been coming to tutorials and office hours, your name is going to be attached to the retake. And you will open that retake. That retake is going to be, again, why do I keep clicking on the wrong class? That retake is going to be there. I'm hoping that the screen has remained here. I'll watch the video and find out in a minute. Asynchronous retake. There's a couple ways to get to it. You can click this folder. When you click this folder, it will take you to another, the original heredity exam. You can see it's obscured. There's a retake here. I'll attach names to it based on those of you who have been coming to the tutorials, who've been coming to office hours. Put your names on there. When you log in, you'll be able to see it. Everybody else will not be able to see it, and you can take the retake. That's it. I'm going to leave you guys be. You guys take care, and I will talk to you guys Wednesday. That's going to be the day I see you guys on Wednesday. Bye-bye.